In this video, we're going to learn about general mechanisms of enzyme catalysis. And the learning objectives are that I want you to be able to recognize in examples each of the four mechanisms of enzyme catalysis. I want you to be able to describe the nature of the interactions necessary for each mechanism. And I want you to understand that a single enzyme can use many of these strategies at the same time. So the first is proximity and orientation effects. If two reactants need to come together to form a product, they have to do so in very specific orientations. They can't just randomly bump into each other and expect the reaction to occur. And what the enzyme can do is that it interacts with each of the reactants in very specific ways so that when they are there within the active site, they are in that active site in just the right orientation such that they can interact properly and form a product. Covalent catalysis is next, and what happens here is that there are transient covalent bonds between the enzyme and the substrate. Now obviously these covalent bonds that are formed must then be broken because an enzyme has to end the reaction in the same state that it began. So let's take a look here at an example. This is chymotrypsin. In blue is the enzyme itself. Okay, and so what we're seeing here is this is the 57th amino acid of the enzyme, that's histidine, the 193rd, that's glycine, and then here's serine. These are the amino acids of the enzyme that are directly interacting with the substrate, and that substrate is a protein itself. Chymotrypsin is a protease. What that means is it cuts off amino acids. And so we see just one part of what's going on here. Here is the protein. This is the substrate of the reaction. Here is the enzyme. And what we have is serine 195 is very close to histidine 57. And look at this interaction here. This nitrogen of histidine 57 is sort of drawing that hydrogen away. And that's making this oxygen very reactive. I've got a question here for you. This interaction between the close relationship between serine and histidine within that enzyme, what do you think that's doing to the pKa of this serine hydroxyl group? Is it making it lower or is it making normal or higher? Well, do you think this serine is going to be able to hold on to that hydrogen better or worse being very close to this nitrogen? It's worse. This nitrogen's drawing it away. That oxygen isn't able to hold on to that protein. And so what's happening here is because of that close proximity of the histidine and the serine, the pKa of that serine's hydroxyl group is going down. It's going to lose that hydrogen. It's going to make this oxygen much more reactive and therefore form a covalent bond with this carbon in the substrate. And that's what we have here. Now this reaction will continue. Okay, and we're not going to show you the rest of it, but what I'm showing you here is this transient covalent bond between the enzyme and the substrate. Okay. Next is acid-base catalysis, and what this is, it's a way that the enzyme can stabilize charges on the transition state. Because if we take a look at a hypothetical transition state, here we have the two substrates, this molecule here, and what they want to do is form a covalent bond here between this oxygen and this carbon, this covalent bond here. The problem is when this happens, we get a transition state in which we get this strange charge on this oxygen, and this nitrogen wants to have a positive charge, and it doesn't have that. So this transition state is very unstable, and we can take a look at these equilibrium arrows, and you notice that it wants to just go straight back to the substrates, right? It's not stable. It wants to go back into the substrates, and we don't want that to happen. We want this to go further and become that final product. This is where an enzyme can help. An enzyme can act as a base to take that hydrogen and act as an acid to add a hydrogen here. Let's see how that can happen. Here's our unstable transition state. Here's that hydrogen we want to remove? Well, we can use a part of an amino acid within the active site to act as a base to grab that hydrogen. Here we want to add a hydrogen, add a proton here. Well, we can use an amino acid within the active site of that enzyme to donate a hydrogen. Okay, that comes there in that active site. And sure enough, after interacting with those amino acids within the active site, we have a stabilized transition state. We no longer have that hydrogen. We finally get that positive charge on that nitrogen. That transition state has been stabilized, has a lower energy, and now that reaction can occur at a greater rate. Now, which amino acids can act as a base to take protons? Which amino acids can act as an acid and donate protons? Well, let's take a look here. Here we have glutamate, aspartate. Both have a carboxyl group. Depending upon the pH, depending upon the microenvironment, they might be able to act as a base and steal a hydrogen, or act as an acid and donate a hydrogen. Let's take a look at another good one. We have, histidine, very common in the active sites of enzymes because its pKa is 6. Can act as an acid to donate this hydrogen, can act as a base to accept a hydrogen here. Okay, so you should be able to recognize instances of acid-base catalysis if I were to give you an example. And finally, metal ion catalysis, and this is very common. About 1/3 of enzymes require metal ions. So here we have these two big balls are magnesium ions. They need them in the active site in order to stabilize that substrate. Sometimes they're used to properly orient the substrate. So here's our substrate here. It's interacting with those magnesium ions in just the right way. Sometimes it stabilizes charges on the substrate. So for example, DNA polymerase is an enzyme that interacts and catalyzes the hydrolysis of DNA. And we know that DNA has a net negative charge, and so we've got these big fat magnesium ions in DNA polymerase to interact with the negatively charged DNA. And then sometimes these metal ions can mediate oxidation reduction reactions, reactions in which we are transferring electrons from one substrate to another. So again, make sure you go through this video and make sure by the end of it that if I were to show you various examples of enzyme catalysis that you can recognize each of the 4 mechanisms, that you can describe the nature of the interactions necessary for each mechanism. Okay, what is it that is necessary about the active site to be able to participate in this type of catalysis? And understand that an enzyme can use several of these strategies. They are not mutually exclusive within an active site of an enzyme.